Welcome to my channel. Today I'm going to deal with the basic steps involved in cloning techniques. This presentation I have divided into three parts. In the first part, I'll be explaining the cloning technique up to the formation of recombinant plasmid. In part 2, I'll be explaining the steps involved in bacterial transformation. And in the last, that is part 3, I'll be explaining about the screening technique. So let's begin with the basic steps involved in cloning technique. Now the traditional cloning technique involves 5 steps. The first step is the vector preparation. The second step is the insert preparation. Then the insert and the vectors are joined together to form a transformed or recombinant plasmid through a process called as ligation. Then this recombinant plasmid is then inserted into the bacterial cell through a process called transformation, and then the screening technique, in which the transformed bacterial cells are identified and cultured. So let's begin with the vector preparation. The cloning vectors are nothing but a DNA molecule that has the ability to replicate in an appropriate host cell, and the DNA fragment to be cloned is inserted into it for cloning. All the vectors that are used for propagation of DNA of interest in a suitable host cells are called cloning vectors. They are also called as DNA vehicles or molecular vehicles. Plasmids are most commonly used as vectors in traditional cloning methods. Based upon their function, plasmids are classified into 5 types. The F plasmid or fertility plasmid. This plasmid contains TRA gene that are capable of conjugation and result in expression of sex pili. Resistant plasmid. These plasmids contain genes that provide resistance against the antibiotics or poisons. Cole plasmid. These plasmids contain genes that code for bacteriocins protein that kill the other bacteria. Degradative plasmid contain genes that enable digestion of unusual substances like taurine, salicylic acid, etc. And the virulent plasmid or virulence plasmid, which turns the bacterium into a pathogen. The word plasmid was first coined by William Hayes and Johannes Lederberg in the year 1952 while they were studying the structure and reproduction of bacterial cells. Plasmids are extrachromosomal, self-replicating, double-stranded, closed and circular DNAs present in the cytoplasm of bacterial cell. The plasmids are ideal vehicles to carry gene because of 2 reasons. First, they can be cut at specific site using restriction enzymes, and the desired DNA can be joined to produce a recombinant. The second reason is they are self-replicating and help in cloning of desired genes. The plasmid vector should have the following three properties. The first property is they should have origin of replication or ORI region. This is a sequence of nucleotide from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cell. The second property is they should have selectable markers or marker genes. The selectable markers help in identify and eliminate non-transformants and permit the growth of selective transformants. The most commonly used selectable markers are the antibiotic resistant genes such as ampicillin, tetracycline, kanamycin, etc. Remember, the normal E. coli cells do not carry any resistant gene against any of these antibiotics. The third property a vector should have is cloning sites. Now, these are the specific sites where the DNA is cut and the gene of interest is inserted into these regions. These cloning sites contain palindromic sequences which are identified by the specific restriction endonuclease enzymes and help in cutting the plasmid. Hence, the RENs are commonly known as the molecular scissors. So, a plasmid should have at least a single restriction enzyme site, or it should have multiple cloning sites. Now, let us see the structure of PBr322 plasmid. Now, this slide shows you a diagrammatic image of the plasmid PBr322. This plasmid was identified by Bolivar and Rodriguez in the year 1977. Hence the name PBR, where P stands for plasmid, B stands for Bolivar, R stands for Rodriguez, and 322 stands for the strain. This plasmid is composed of 4,361 base pairs. It has got a ORI region, 2 selectable markers, one in form of ampicillin resistant gene and the second is in form of the tetracycline resistant gene, and then it has almost 13 multiple cloning sites, but of which 7 are of importance. Now this EcoR, this is the region where the REN EcoR can cut it. Now PUV1 and PST1, these are the REN, that is the restriction enzymes that can be inserted in this ampicillin resistant gene. Then in the tetracycline resistant gene, there are 3 restriction sites. One is cut by BMH1, SAL1 and XMA3. And here this is a ROP gene, that is the resistance osmotic pressure gene, where this PVU2 restriction site is present. Now this is a diagrammatic representation of the plasmid PuC18. This plasmid was discovered by J. Messing and Vieira from University of California in the year 1985. They are the most popular plasmid-based vectors and contain most of the desirable features for day-to-day cloning experiments. Now here, the P stands for plasmid, U stands for university, and C stands for California, and 18 stands for the strain. Now the unique feature of this plasmid is in the LAC-ZERT gene, it has got these multiple cloning sites, and this unique feature is used in the blue and white screening technique, which I'll be explaining to you in the part 3 part. So this is PuC 18 plasmid. It is composed of 2686 base pairs. It is having a single OR aeration. Now this is a lactose operating gene, that is a lac operon gene. Now here this is ampicillin resistant gene, and here it has got this the OR region. Now with this background of plasmids, let us now see the different techniques involved in vector preparation. The first step in vector preparation is to create an insertion site by restriction digestion. Now this restriction digestion is done by the restriction endonuclease enzyme. It's commonly called as REN, and because it is used in cutting the DNA, it is also known as molecular scissor. The first REN, that is HIND3, was discovered by Hamilton Smith in the year 1970 in the bacterium Haemophilus influenzae. At present, more than 900 restriction endonuclease enzymes have been isolated from more than 230 bacterial strains. Now, there are certain principles involved in the naming of restriction endonuclease enzyme. The restriction endonuclease enzyme is represented by a 3-letter code. The first letter refers to the genus name of the bacteria from which it is derived and is written in capital. The subsequent 2 letters represent the species name. The 3 letters are generally written in italics. The subsequent letters represent the strain, and the Roman number indicates the different REN derived from the same organism. For example, now here we have this 2 restriction endonuclease enzymes, EcoR1, EcoR2. Now here the E, capital letter E, stands for the genus name of the bacterium Escherichia, and the small letter CO stands for the species name coli, and R stands for the strain RY13, and the Roman letter number 1 and 2 stand for the different RENs extracted from the so on. Now, these restriction endonuclease enzymes cut DNA at specific palindromic sequences. A palindromic sequence is a sequence of nucleotide bases on DNA that read the same both in forward and backward direction. Now, for example, here you see this is a DNA strand. In the forward direction, the nucleotides read as GCATGC. And in the backward direction, that is the opposite strand, in the reverse order also they read as same GCATGC. So such sequences are called as palindromic sequences. The RENs are very specific in recognizing their palindromic sequence. For example, EcoR1 recognizes the palindromic sequence GAATTC. So this palindromic sequence is identified by the restriction endonuclease enzyme EcoR1, and it cuts the DNA at the G site, that is the phosphodiester bond between the G and A nucleotide from 5' in the forward direction and the 5' in the backward direction. The phosphodiester bonds are cleaved and then the DNA is cut and releasing these free nucleotides. These free nucleotides are called sticky ends, and it is in these regions our gene of interest is inserted. So this is how the palindromic sequences are identified by these specific RENs and help in cutting the DNA. So now let's see the whole concept of the vector preparation. Now here this is a test tube containing the extract which contains the plasmids isolated from bacterial cell. So here we have isolated these plasmids. Now this is the diagrammatic representation of all the different types of plasmid that are present in the extract. Now can you identify, looking at the structure, which plasmid it is? So now to this extract of plasmid we add the REN. So here we are using the EcoR1, that is the restriction endonuclease enzyme extracted from the Escherichia coli RY13 strain. Now, as I've told you earlier, this EcoR1 enzyme identifies the palindromic sequence GAATTC. So, once the palindromic sequence is identified, the phosphodiester bonds between the guanine and adenine in the opposite strand and guanine and adenine in the forward strand are cleaved, and then the DNA is separated, and after separation of the DNA the sticky ends are formed. Now if it is left in this way, there is every chance that again the sticky ends may join to each other based upon complementary pairing. Hence, after restriction digestion, dephosphorylation of the vector is necessary. So this dephosphorylation of the vector is done by using an enzyme phosphatase, and this phosphatase enzyme removes the phosphate attached to the adenine nucleotide here, adenine nucleotide here. Now once the phosphate is removed, then there is no chance of the formation of the phosphodiester bonds. Hence, the cut ends remain separated. So this is how the vectors are prepared. So now the culture is containing all the plasmid with cut ends and the free sticky ends are present. Now we shall see the next part, that is insert preparation. The source of insert for cloning may be a genomic DNA, a portion of another plasmid, or a linear DNA fragment. Regardless of the type of source DNA, a common first step in preparation of insert is to perform restriction digestion to generate compatible ends for subsequent splicing into the vector. As with vector preparation, restriction enzymes that are suitable for cloning of the insert into the vectors are selected. Now, one of the most popular strategy is to perform double digest of both the insert. So there are three types of insert preparation. The first step is the double digest using two enzymes. Now here the vector also is cleaved in the forward strand by the same enzyme, and the other end is cleaved by the other enzyme. So in this way, what happens is, when such two enzymes are used, the orientation of the required gene is maintained. That is the advantage of this two enzyme with the double digest technique. So here you see, now here this end of the vector is digested with EcoR, and this end of the vector also is digested by EcoR. Now here the other end, here we are using a different restriction endonuclease enzyme. Here we have used KPNL. So KPNL cuts the palindromic sequence GGT-AG. So here this KPNL used in the insert also at this end and the vector also in this end. So because of such complementary pairing, the gene of interest, the orientation is maintained and is inserted here, and you get a ligated plasmid. So this is one technique. Now the second is single digest using one enzyme. So this is the most common type of the insert preparation, insert and vector preparation. And in this presentation also, I am using the same technique of insert preparation, where we are using a single enzyme. Now the same enzyme is used in both the vector slicing and insert slicing. So we have the complementary pairing at both the ends here, and then it is inserted into the plasmid. Now the third is single digest using two enzymes. Now in situations where it is not possible to use a single restriction enzyme, a pair of enzymes that have different recognition sequences but generate compatible overhangs can be considered as an alternative. For example, BAMH1 recognizes GGA-TCC palindromic sequence, and BGL 2 recognizes AGATCC. Both generate GATC overhangs. I can see here GATC overhangs here, GATC overhangs here. Okay, so here two different enzymes are used, but vector is digested with the different enzyme and the insert is digested with the different enzymes, but both are giving rise to the same complementary overhangs. So this technique is known as single digest two enzyme technique. So once the fragments of interest are obtained, a ligation reaction can be set up to join the insert and the vector. The most common enzyme used for ligation is T4 DNA ligase, which links DNA ends between the 5' phosphate and 3' OH group. The T4 DNA ligase reaction requires ATP, DTT, and magnesium divalent cations as activators, which are generally supplied in the reaction buffer. So here in the test tube, we have the plasmid with sticky ends. Now in that test tube, we add the DNA fragments, that is the insert. Now here you can see both the vector and the insert are digested by the same enzyme, that is EcoR. So we have this AATT ends here, AATT ends here. So this is a single digest, single enzyme technique where both the insert and the plasmid are digested by the same endonuclease enzyme. So then we add the enzyme T4 DNA ligase. So now this DNA fragment to be cloned comes and attaches at the sticky ends, and the sticky ends are joined together by the T4 DNA ligase enzyme, and that results in formation of a plasmid with the required or foreign gene. So you can see this is a plasmid. Now this was the original plasmid. Now here this green colored segment is our gene of interest, which we have inserted into the plasmid. So such a plasmid we call it as recombinant plasmid. So here I conclude this part 1, where we have seen how the vectors are prepared, how the inserts are prepared, and how the insert and the vectors are joined together, resulting in formation of the recombinant plasmid. Thank you. And in part 2, we'll be seeing about the bacterial transformation.